You know, every single thing about you, from the color of your eyes to how you fight off a cold, it's all written in this incredible molecular code. We're talking about DNA, the blueprint that makes you, well, you. So how on earth does it all work? Let's dive in and find out.
Seriously, just stop and think about that for a second. Every part of you, from your brain to your big toe, is built from a set of instructions packed into a space that's literally too small to see. I mean, how is that even possible? How can something so ridiculously complex be stored so compactly? Well, the answer, it turns out, is this surprisingly elegant concept known as the central dogma.
Okay, so this right here, this is the master plan for pretty much all of life. The genetic information flows from the master blueprint, the DNA, to a temporary working copy, which is RNA. Then, that copy is used to build the actual machinery of life itself, proteins. DNA to RNA to protein. That simple three-step flow is the roadmap for everything we're about to cover.
All right, so if DNA is the blueprint, we first need to understand the material it's written on, right? Let's take a closer look at the physical structure of DNA itself.
Get ready for this because this is mind-blowing. If you were to take all the DNA from just one of your cells and stretch it out end to end, it would be over 2 meters long. That's taller than most people, and that's from a single microscopic cell.
To put that into perspective, the nucleus of a cell is microscopic, so cramming 2 meters of DNA inside it is the equivalent of trying to stuff 25 miles of superfine thread into a tennis ball. It's just an incredible feat of biological engineering, and it's all done by coiling and supercoiling the DNA strand with this, mind-boggling efficiency.
Of course, the iconic shape of DNA is the double helix. Just imagine a twisted ladder. The sides of the ladder, they're made of sugar and phosphate, and they provide the structure. But the magic, the actual information, it's all in the rungs of that ladder.
So these rungs are made of just 4 chemical bases, A, T, C, and G. But here's the beautiful, unbreakable rule. A always pairs with T, and C always pairs with G. It's that simple. This pairing, this complementarity, is the entire secret to how DNA can store and copy information so perfectly, generation after generation. So we have this incredible, tightly packed code, but a blueprint is pretty useless if no one can read it. So how does a cell actually use this information to build all the machinery of life? Okay, so before a cell can divide, it has to make a perfect copy of its DNA. To do that, the helix unwinds, splitting right down the middle like a zipper. And because of that base pairing rule we just talked about, each separated strand becomes a perfect template. New bases just lock right into place, building a new strand alongside each old one, and boom, you have 2 identical DNA molecules where you used to have just one.
But a cell doesn't need the whole blueprint all the time, right? When it needs to build a specific protein, it performs transcription. The best way to think of it is like photocopying a single recipe from a giant cookbook. A small section of DNA, we call that a gene, unwinds, and a messenger molecule, mRNA, makes a quick copy of just that one recipe and carries it out of the nucleus.
Now this mRNA message is written in that 4-letter language, but it's read in 3-letter words called codons. Think of them as the words in the recipe. Each codon corresponds to one of the 20 different amino acids, which are the fundamental building blocks of proteins. So a sequence like AUG, for example, is the codon for the amino acid methionine.
But wait, how does the cell translate the codon language into the protein language? It needs an interpreter, and that interpreter is called transfer RNA, or tRNA. Each tRNA molecule is like a specialized adapter. It's built to recognize one specific codon on the mRNA, and it carries the one and only one amino acid that matches it.
And this all comes together in the final step, translation. The mRNA message travels to a cellular factory called a ribosome. As the ribosome moves along the mRNA, reading the codons one by one, the correct tRNA molecules shuttle in, dropping off their matching amino acids. The ribosome links these acids together into a long chain, which then folds up into a complex, functional protein. This whole process just brilliantly shows the central dogma in action, from DNA to RNA to a finished protein.
So, understanding this fundamental process has unlocked some absolutely incredible technologies. Let's look at how we've applied this knowledge to read our own genetic code and what we can actually do with that information.
Back in 1990, scientists embarked on one of the most ambitious projects in human history, to sequence the entire human genome. This was a massive international effort, kind of a moonshot for biology, and it took 13 years and billions of dollars to complete.
And the scale was just staggering. The goal was to figure out the precise sequence of all 3 billion chemical base pairs that make up human DNA. To put that in perspective, if you printed it out in books, it would fill a pretty big library.
You know, one of the most shocking discoveries was that less than 2% of our entire genome actually codes for proteins. A huge chunk of it is these repetitive sequences we don't fully understand. And for over half of our genes, the function is still a complete mystery. It showed us that what we used to call junk DNA might actually hold the keys to understanding the very complexity of being human.
So, the project also confirmed that all humans are 99.9% genetically identical. And that raises a fascinating question, doesn't it? If we're all so similar, how in the world does forensics use DNA to uniquely identify a single person out of billions? Well, the key isn't in the 99.9% of genes that make us human, but in that tiny fraction that makes us unique. It turns out those non-coding repetitive stretches of DNA vary wildly from person to person. That little 0.1% difference is where your unique genetic identity is found.
So what forensic scientists do is create a DNA fingerprint by analyzing the length of these repeating segments at several different locations across the genome. This creates a really unique pattern of bands, almost like a genetic barcode. When the pattern from a crime scene sample is a perfect match to a suspect's, like you can see here with suspect B, the connection is pretty much unmistakable.
And this all circles back to that beautiful, simple discovery from 1953. When Watson and Crick published their paper on the double helix, they included this sentence, which is now famous for being one of the greatest understatements in the history of science. They instantly saw that the very structure they had discovered, that simple 8T-CG pairing rule, was the secret to how life copies itself. It's just brilliant.
And that brings us to today. We've moved from simply reading the code to actively writing and editing it with powerful technologies like CRISPR. This knowledge of DNA has given us immense power over the fundamental building blocks of life itself. And so, the profound question for our future is no longer can we, but should we?